Okay, guys, so we are going to move on to DNA replication and how DNA makes more of itself. So, the functions of a cell are determined by its DNA, and cells have to reproduce many times in complex organisms, make trillions of copies from one original cell. But when the cells reproduce, they must replicate or copy their DNA. The structure of DNA reveals how trillions of copies of DNA in one of your cells can be made and be almost exactly the same every time. It's pretty amazing. And one of the things that makes DNA so amazing is the fact that it has this ability to self replicate. So, Watson and Crick published the structure of DNA in a short article in 53. They stated it has not escaped our notice that the specific pairings we have postulated immediately suggest a possible copying mechanism for the genetic material. And the fact that there are two DNA strands that are mirror images for one another suggests how copies could be made in DNA sequence. So each molecule of DNA is made from a template of a new strand, a template and a new strand. So what happens is the DNA molecule will unzip and you'll leave one old strand, and then a new bit will come in and make from there. So you always have a old strand and a new strand in every piece of DNA. It's called semi-conservative. It keeps some of the old information, well, it's all the same information, but it keeps some of the old um, copies versus some of the new copies. The template strand is the parent strand, and since it came from the original, and the new strand is the daughter strand. Okay, so the template strand of the DNA molecule separates. The new strands are made on the inside. So it separates, it gets unzipped. We'll talk about how in a minute. And then the new copies will go next to this. So you can tell this one is the leading strand because it's nice and smooth. The green arrow doesn't have any breaks. So as it unzips, now we'll put a G, now we'll put an A, now we'll put a G, and it'll just keep copying all the way up. This one is going to be the lagging strand. It's the lagging strand because it has to unzip and then it starts copying, and then unzips and it starts copying, making these fragments, also known to be Okazaki fragments. There is an animation here. If you can take the time to open that up and click on it, please, it'll help understand this. All right, so to add new nucleotides, they are only added to the OH end, the 3' end, and not the 5' end. So that's why we get the leading and lagging strand. On the one that is got the 5' up, then we will not be able to add. On the ones that have the 3' up, then we can add, and that'll be the leading strand. So DNA nucleotide monomers are made ahead of time and stored in the cell. They're just floating around, ready to go, ready to be reused when they're ready. DNA polymerase is going to be the enzyme responsible for picking this up and putting it where it matches. So blue pairs with white. So in this case, this one will be ready. DNA polymerase will come in and move that into place. So it is semi-conservative. That's because again, it'll be unzipped and one old will be in one place and one old will be in another place. And then we'll have the daughter cells in both places. So the 3' end of the strand has a phosphate at the end. True or false? Oh, sorry. False, because it has the OH end. Good. Why does the DNA strand only grow in the 5' to 3' direction? Because the DNA can only add nucleotides to the 3' end of the molecule. Because the DNA can only add nucleotides to the 5' end of the molecule. Because mRNA can only read DNA from 5' to 3'. Because mRNA can only read DNA from 3' to 5', or I need help. Did you say? Good. Because it only is going to add to the 3' end, so it has to unzip from the 5' to the 3', opening up that end, that 3' end. All right, so let's practice this. I am not going to be able to draw, so we will just discuss. Take a second and see if you can make the copies here. Okay, so pairing up with A is going to be T. A, G, C, 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 A, T, T, G, C, G, C, A, T, T, T. And notice on both of these, we went from 5' to 3'. We're always adding to that 3' end. Same thing going on down here. So let's go ahead and copy that one. C, C, A, T, G, A, T, T, A, G, C, T, C, G, 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 A. All right? So make sure you can copy those. You do the opposites. Again, A always pairs with T and G always pairs with C, and we always add onto the 5', or I'm sorry, the 3' end. If the parent DNA strand is 5' to 3', what will the daughter strand be? And so take a second and get your answer. Did you say D? Good. First of all, we know we need it to start with 3', so it is anti-parallel. This one's 5', so the next one needs to be the opposite, 3' to 5'. And then it will be opposite, not the same. And it is DNA, not mRNA, so we do not need the U's. So D is the best answer. A could have been a good answer if these were changed, if the direction was changed. B is not a good answer because it is the exact same. C is not a good answer because it's an RNA. D is the best answer. Okay, so the molecular process of replication. The strand of DNA is replicated in segments at intervals down the DNA molecule. Portions of the two strands separate, creating replication bubbles. Either side of the replication bubble is known as a replication fork. So, and that they will be replicating in different parts of one molecule all the time. So here's replication fork, here's replication fork, here's replication fork, here's replication fork. And so as they open up, you have the 5' on one side here and the 3' on the other side here, and you'll be making new. Okay. So at the replication fork, there's a lot of processes that help make sure mistakes don't happen. It's actually a really amazing proofreading system because if you made trillions of copies, chances are you'd make way more problems than DNA ever has. And any change in the nucleotide is going to be a mutation. So a couple things to note here. They have a helicase. The helicase is going to be the unzipping enzyme. It comes in, it unzips the two strands. You have single-stranded binding proteins. These just help keep things apart. So that way they don't want to come back together. You have DNA primase, which is going to be the primer for the lagging strand. It's going to like hold a placement so that way it's like, okay, now I'm going to add, now I'm going to add, now I'm going to add. Like this is my starting point as it unzips more. And then the DNA primase will pop onto another spot and start adding again. An RNA primer is going to be the thing that starts it on the RNA side. And then DNA ligase is going to be the thing that actually glues together any of your Okazaki fragments. So any of these spaces that are here, the DNA ligase will come in and glue those back together. So it's one continuous strand. So at the end, they don't look any different. In the middle, there's going to be these little chunks, but at the end, there won't be. And then there's DNA polymerase. DNA polymerase is what is actually making the copy to put on there. So, and on the leading strand, it is much less complicated. Still has the single strand of binding proteins, but only has DNA polymerase. It does not need the DNA ligase. It does not need the DNA primer or the RNA primer to make this work. It just needs these two things. Okay. Okay, topoisomerase. There we go. Sorry, guys. Is an enzyme that controls the winding and unwinding of DNA during replication. So doing any of this would be really difficult if it was still wanting to twist. And anything that's always been twisted, you know, it wants to twist back up. It's like wrinkles. They don't want to go back to normal without something to make them. Topoisomerase is going to be the thing that helps them. So a winding problem in DNA arises due to the intertwined nature of the double helix structure. During DNA replication, DNA becomes overwound ahead of the replication fork. This tension would eventually stop replication, but topoisomerase will not let that happen. Helicase is the unzipper. It breaks open the weak hydrogen bonds that holds them together, opening both sides so it can become the new templates. The single-stranded binding proteins, or SSBPs, sometimes known as, are little binding proteins that help them not come back together after the helicase. Leading versus lagging strand. Again, the leading strand is going to be where you are going to be able to add to the 3' end easily. The lagging strand is where you have to add to the 3' end in segments. So the leading strand for DNA polymerase is very easy. Regarding to the template that is oriented 3' to 5', DNA polymerase can follow behind the helicase and read from the 3' to 5' direction, synthesizing the new leading strands from the 5' to the 3' direction. So it's constantly making new in the right direction, no problems. So that one is much easier. Now on the lagging strand, that is not easy. The DNA polymerase can only read from the 3' to 5' direction. So the template strands running from the 5' to the 3' is new. The lagging strand has to be made in reverse. This starts with the enzyme called primase that adds RNA nucleotides as a primer for the DNA polymerase. It puts these little primers on here and says, okay, we're ready to go ahead and do this from here on out. And so it makes it in those directions. DNA polymerase can latch onto the RNA primers and begin to write the new fragments on the new strand. Since it's going away from the replication fork, it only does a portion, and then it jumps back and does another portion. So Okazaki fragments are what you get in that case, these little holes. And when the RNA primers fall away, then you have these little holes. So then the DNA primase, ligase will come in and they will fix that. So ligase is going to be the enzyme that fills the gaps between the Okazaki fragments. Now, let's see if you remember any of that. Hydrogen bonds hold base pairs together in DNA. Which enzyme severs these bonds during replication? Ligase, polymerase, primase, helicase, or I need help? Did you say helicase? Good. A mutation has caused a change in the shape of topoisomerase enzyme. This would mostly affect the directionality of DNA synthesis, the formation of bond between Okazaki fragments, the tension in the DNA double helix, the separation of DNA strands, or I need help. You say tension? Good. Yeah, without that there to help unwind it, it's just going to get tighter and tighter, and eventually it will snap. Incorrect base pairing does occur during replication. However, most of these errors are corrected. Which enzyme effectively decreases the mutation rate? Helicase, ligase, primase, or polymerase? Which one do you think will help fix that? Did you say polymerase? Yeah. Polymerase is the one that's making the copies, bringing the right parts to where it needs to be. So it's in charge of making sure there's no typos. Now, PCR replication is really super important. PCR replication is polymerase chain reaction. polymerase chain reaction is a way of taking a small amount of DNA and copying it and making lots and lots and lots of copies so we can test it for manipulation. So it's pretty much using the principles of DNA replication to make this happen. It's carried out by a machine, you see there, that utilizes repeating cycles of heat. So it's going to heat up and cool down and heat up and cool down. DNA polymerase, DNA's primers, and free nucleotides will build onto the new copies of the DNA fragment. This allows us to take a small amount of DNA and turn it into a large amount. So this is one way that if they found some DNA underneath somebody's fingernails in an assault victim, they can take that little bit and copy it to where they have enough to actually see the whole picture. If otherwise just looking at one tiny little fragment would be hard, but if you copy it enough, then you can actually see it. It's how they can take your swab for a COVID test and find if there's any COVID DNA in there, because they're going to find the base pairs on the gel electrophoresis for the amount. They're going to isolate that, and then they're going to copy a bunch of those so they can see if there's any there. All right, so PCR replication. DNA is heated to a high temperature, and when that happens, the DNA splits. And that represents, so that separates the double helix. So it's kind of like helicase in that case. We're heating it up to separate it. Then as it cools, the primers and the polymerase will actually attach anneal onto there. So as it cools in cycle 1, now the primers and polymerases are on there. Then the temperature is increased a little bit, not as much as it was in number 1, to increase the rate of replication. So now it's just warm enough that it's going to start copy, 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 copy. And then in cycle 2, it happens again. But with all of these, now they make another copy. And then in cycle 3, it happens again. But each of those are now going to make a copy. So it's going to be in this logarithmic rate. Anyway, you know what I'm trying to say. I'm sorry, guys. Anyway, so it just keeps expanding exponentially like that. A single DNA molecule is placed in a PCR machine. After 20 cycles, how many copies of DNA will be present? 1,048,576. Did you get that? It's a lot, right? This is an easy way. So 20 cycles is pretty common. It's an easy way for them to really be able to get that whole picture. The Taq polymerase is a typically used in polymerase chain reactions. This polymerase enzyme is found in thermophilic bacteria, Thermos aquaticus. What is the best explanation for using this enzyme? Why would they want to use this Thermos aquaticus? Enzymes from the thermophilic bacteria are stable at high temperatures. Bacterial enzymes are easier to replicate than eukaryotic enzymes. Ethical objections exist to use human macromolecules. Only Taq polymerase is available commercially. Did you say A? Yes, because it is a thermophilic bacteria, it is a heat-loving bacteria, thermophilic, heat-loving. So it has already stuff in it to counteract being broken down by that high temperature. So it only leaves the DNA that would be separated, and the polymerase is going to be left intact because it is from an area that likes the high temperature. That'll lead us to transcription and translation, and we are going to pick up transcription and translation in the next video. So in this video, we learned about DNA replication. DNA replication is all about unzipping the molecule and then putting new on. To keep it from twisting, remember that's topoisomerase. To unzip it is helicase. You have then the single-stranded binding proteins to keep it from coming back together, keep it separated. And then you have your RNA, your DNA primase for the lagging strand, which will put the RNA primers on there, and then it'll add to the 3' end. But then at the end, that RNA primase will leave, and we will need to ligate the two pieces, the Okazaki fragments, together. On the lagging or leading strand, it's much easier. As it unzips, the DNA polymerase can just make new in the proper direction, so it keeps going. And so that one is much more smooth and much more quick. And that's it. That's the how DNA copies itself. So take a time. Let me know if you have any questions, and we'll go from there. All right, guys, have a great day.